School of Medicine with topic the artificial drug screening system development from the cardiovascular disease. And from our beloved lecturer, Dr. Wawaimuli Rozali, with topic the recent clinical relevance and translational limitation of animal models and cardiovascular medicine. We will be guided by Dr. Agian Jeffilano Barinda or Dr. Jeffy as our moderator. We will listen to the interview, which will open the participant side in biomedicine. Okay, before we continue, I would like to read about the webinar rules, which okay, while we wait the admin to uh, present the webinar rules. Uh, okay, I would like to read about the webinar rules first. Participants should present good manner during the event, and participants should have their microphone in muted condition, and participants are allowed to ask questions through Zoom chat by mentioning name and your institution. And participants are required to fill in attendance link twice to get webinar certificate. And the attendance link will be posted 15 minutes after the opening and 15 minutes before the closing. Okay. This webinar would not have been possible without the support of the PBS as the institution. For that, let's listen to remarks from the head of study program, Professor Asmarina, Magister of Science. So, Professor Rina, would you please have a word from us?
Okay. Okay, thank you, Dr. Oka, for the introduction for this webinar today. So, good morning for everybody here. I'm Asmarina, as acting of doctoral program in Biomedical Sciences, Faculty Medicine, Universitas Indonesia. First of all, I would like to welcome you all to our guest lecture today that will be delivered by Professor Shinsuke Yuasa from Keio University, Japan. And yeah, of course, we are very grateful to Professor Shinsuke Yuasa for the willingness also to become our visiting professor in our program. And today we'll give a lecture for our staff and students. I would also express my appreciation to Dr. Wawaimuli, who arranged this event, and as well as to the Dr. Jeffy that has introduced the Professor Yuasa to our faculty last year, and then today is the realization of our meeting in last year to make a collaboration. So this event is actually supported by our university, Universitas Indonesia. Professor Yuasa, we would like to give a special grade as a visiting professor in our program. This program, visiting professor from abroad scientists, has aimed to increase the knowledge of our student and our staff. So as well also to make a joint research. With this visiting professor, we would also to make this professor in university all over the world, abroad, to become a supervisor also for our student, and also as examiner for our examination for our student. So I think that's all for my introduction or in this opening speech today. But I think now we can begin the seminar today, and we hope that this material or the knowledge from this webinar can make increase our the knowledge of for our student and also for our staff. Yeah. And then we hope also with this seminar we can continue with the research collaboration, yeah, with the Dr. Wawaimuli, for example, also with Dr. Jeffy, yeah, in this field, yeah, in cardiology, yeah. Okay, that's all, Dr. Oka. Thank you, and yeah, nice in this meeting, yeah. So
Okay.
thank you very much, especially for Professor Yuasa for your meeting today.
Yeah.
Today.
Thank you very much, Prof. Rina, for the delightful opening. Okay. Now, we will continue to our main section, which is topic presentation. Therefore, let me invite Dr. Agian Jeffilano Barinda, Ph.D., as our moderator, who will guide our discussion in this morning. Okay, Dr. Jeffy.
Okay, thank you very much, Dr. Oka. So, good afternoon in Indonesia and also in Japan time. So, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Please allow me to introduce myself. My name is Jeffy from Pharmacology Department, Faculty of Medicine, Universitas Indonesia. So thank you very much. It's a great pleasure for me to be a moderator in this session. So today we will have two honorable speakers, the Dr. Yuasa from the Department of Cardiology, Keio University from Japan, and also Dr. Wawaimuli Arozal from the Pharmacology Department, Universitas Indonesia. So first of all, so I would like to, we will have a lecture from Dr. Yuasa at the first time. So please let me briefly introduce Dr. Yuasa. So Dr. Yuasa is an assistant professor of the Department Cardiology in the Keio University in Japan. So he had both of the M.D. and Ph.D. in the Keio University, and currently he is also a member of the American Heart Association's Japanese Circulation Society and also Japanese Society of Regenerative Medicine. So he has been awarded as a Young Investigator Awards in the Japanese Circulation Society 2008, Lewis and Katz of the Basic Science in the Young Investigator in American Heart Association 2008, Young Investigator Award in Japanese Society of Regenerative Medicine 2011, and also Young Investigator Awards in the Korean Cardiovascular Medicine in 2013. So he has several publications in the reputable journals. For example, the 2 times in the Cell Stem Cells as a first authors in the 2010, and also in the Circulation Research as a first author also in the 2011, and also he has many papers in the Nature Communication, PNAS, Cell Metabolism, Nature Protocol, and also Circulation as a co-author. So today he will give a talk about the artificial intelligence-based drug screening system development for cardiovascular disease. So he is cardiologist. He also expertise in the iPS cell-derived cardiovascular and also expertise in the artificial intelligence. So it will be interesting to hear his excellent lecture. So if you are ready, Dr. Yuasa, so you can start the lecture from now. Thank you very much. Please, Dr. Yuasa.
Thank you, Jeffrey. Can you hear?
Yes, please.
Yeah. Okay, thank you.
Thank you for invitation to this guest lecture in University Indonesia. And actually, last year I was there, and in this situation I cannot go abroad, and so, but it is easy to connect by web conference, so it's very convenient.
Yeah.
So today I will introduce our research finding about AI-based drug screening system development for cardiovascular disease. I am Shinsuke Yuasa from Keio University. So Keio University is in the Tokyo, so about 6,000 kilometers from Jakarta. And so, and Tokyo is here, and Osaka is here, and Niigata is here. So, and this is Yamanote line and central line of the train. And so this is Tokyo, and this is Shinjuku, and this is Keio University. So about 3 minutes from Keio University, there is Japan National Museum, National Stadium. This is the main stadium for the Olympic Games next year. I don't know Olympic Game will held or not. This is a stadium, and this is Keio University. And also, about 5 minutes walking from university, there is a big national garden, Shinjuku Gyoen Garden. And this is a Shinjuku Gyoen Garden. In spring, and cherry blossom is very beautiful. And then from Keio University, there are many places. Shibuya, Shinjuku, Harajuku, about 10 minutes by train. And this is Shinjuku, and this is Keio University. So Akihabara, Asakusa, and Tokyo Disneyland is very close to our hospital and our laboratory. But actually, we cannot go many places because we have to do our research. Anyway, so move on the study. So this slide shows the World Health Organization study, global burden disease study. It provides an update snapshots of the state of cardiovascular disease, reaffirms that it continues to be a significant public health concern worldwide, despite impressive advances in technical capacity for preventing and treating cardiovascular disease. About 400 million patients with cardiovascular disease worldwide in 2016. So many patients suffering from cardiovascular disease. One of the suggestions from global burden disease study is for countries to consider further investment in cardiovascular disease study towards reducing the burden of cardiovascular disease. Despite the global burden of cardiovascular disease, investment in cardiovascular drug development was decreased over the past 2 decades because we think there are several limitations. Research technology for developing novel drugs is limited, and also the disease model examining cardiovascular disease is also limited. Success rate is quite low to develop a novel cardiovascular drug. The high cost of conducting cardiovascular outcome trials in humans. So to develop a novel drug with a high success rate, we should apply new technology to cardiovascular research. So we focused on, at first, iPS cells. In 2006, Dr. Yamanaka in Kyoto University firstly reported mouse iPS cells can be derived from mouse somatic cells. In 2007, next year, human iPS cell can be generated from human somatic cells. In 2012, he received a Nobel Prize, and this is a press release from Nobel Committee members. So they say iPS cell can be generated from humans, including patients with disease. Mature cells, including nerve, heart, and liver cells, can be derived from these iPS cells, thereby allowing scientists to study this mechanism in new ways. We obtained somatic cells from patients. Usually we use blood cells. Then we transfer and then we generate iPS cells. Then iPS cells can differentiate into any type of cells, nerve, heart, and liver cells. Using those cells, we can conduct a regenerative therapy and also disease modeling and drug discovery. So this slide shows a flowchart of potential application of iPS cells. We conducted many studies to use iPS cells for the clinical trials. So we generate many iPSC generation method and also cardiomyocyte differentiation method and purification method. And then we plan to regenerative therapy in next year or next year. Also, we try to elucidate disease mechanism in cell culture dish as a disease modeling, and also we conduct drug screening. So actually, initial step of the IPS study is differentiation and microscopic observation. iPS cells can differentiate into any type of cells, including cardiomyocytes and endothelial cells. And then we firstly observed through microscopy. This is a phase contrast image of iPS cell-derived cells. And so we identify this is a cell, but we cannot identify what type of cell this is. So to identify differentiated cells, so cells are usually visualized by molecular technique immunostaining. So to identify what type of cells, we usually perform immunostaining by using cell surface antigen. And then primary antibody, secondary antibody, and fluorescence is used for the visualization. CD31 is an endothelial cell marker, so we identify this is endothelial cell. This is the same visual field. This is endothelial cells, but this is not endothelial cells. From this image, we cannot identify the difference. FACS analysis clearly shows there is about 30% of cells are endothelial cells. But to use iPS cell-derived cell for research and clinics, any labeling technique should not be used. So this is our question. Hepatocyte, cardiomyocyte, neurons. Each cell type shows identical gene expression pattern, including structural protein, which suggests that each cell type would have unique morphological feature. So different shape. So can we identify individual differentiated cells by phase contrast image without labeling? So recently, AI-based image recognition system is greatly improved. This is a press release from NEC company in 2017. So NEC facial recognition was adopted by major city across Georgia company. As you can see here, this system can identify the human face like this. And also among them, they also can identify VRP and blacklist person. So this system is widely accepted in the major country. NEC's facial recognition software uses deep learning technology and can identify individuals via low resolution face image captured by a distant camera. So we think this is a similar situation with us. So we can identify this is cells, but we cannot identify what type of cells. So therefore, human cannot identify cells by microscopy without labeling, without immunostaining, but machine can identify the cell types. So to use machine learning technique, we have to know the running algorithm. Machine learning tasks are classified into several broad categories. The types of machine learning algorithms differ in the approach, the type of data they input and output, and the type of task or problems that they are intended to solve. There are three major algorithms, such as supervised learning, unsupervised learning, and reinforcement learning. So among them, we focus on supervised learning.
Sorry, someone, the participant, so please mute. Okay, thank you. Okay, please continue, Dr. Yuasa.
So supervised learning is the machine learning task of learning a function that maps an input to output based on example input-output pairs. So this is an example to identify cat's face. At first, we prepared the cat's face like this, and then put into the machine, and the machine ran to identify cat's face. After the network is learned, and then we prepared many animals' face, cats, dog, like this, we put into the machine, and the machine can identify cat's face or not cats. So this is supervised learning. So next, we move on the iPS cell-derived endothelial cell recognition system by machine learning. So for supervised learning, a labeled image of endothelial cells are prepared and put into machine. And trained network can identify the cell type. Endothelial cells are not endothelial cells. So this is a study design. At first, we define the target block. This is about one cell size, an image area to be predicted. And this is endothelial cells or not endothelial cells. So we also prepare reference block. A little bit larger image of same size, we can change the size. And this reference block is an image area used for prediction. So reference block is an input image, and image is input into neural network, and then neural network predict this is endothelial cells and not endothelial cells. Now compared with the answer, neural network gets smarter. So this is a standard protocol of endothelial cell differentiation from human iPS cells. At first, we induced a misnomer specification by cytokine administration. And then at day 4, we induced endothelial cell specification by adding other cytokine. At day 6, we analyze like this. There are so many CD31 positive endothelial cells like this, about 37%. But in each experiment, differentiation efficiency is also variable, and sometimes high, sometimes low, about 20% to 40%. So in each experiment, we have to identify the efficiency of endothelial cell differentiation. So next, we also have to create the answer set. To create answer set, at first we take phase contrast image and define the target block, and after immunostaining, this is important to obtain the image in same visual field, and then after noise reduction, shading correction, and the binarization, we obtained like this image, black and white color image. So we define the threshold, white pixel to black pixel ratio. Over 80% of white pixel, we define this is endothelial cells. Under 80%, this is not endothelial cells. So this is our defined threshold, and the answer is 0 or 1. So this is outline of machine learning strategy. At first, obtain phase contrast image and define target block and reference block. Reference block is input into the neural network, and neural network predict this is endothelial cells or not endothelial cells. After immunostaining and binarization, and we focus on target block and compared with the threshold, answer is created. Endothelial cells and not endothelial cells. Compare the answer and prediction, and then network is adjusted. And finally, train network answer, this is endothelial cells or not endothelial cells. This is the results of past trials. In this case, reference block is same size as target block. This is the result. This axis is the number of image we used, and also this axis is the F1 score, and accuracy is the accurate marker of the machine learning. So as you can see here, the accuracy of endothelial cell identification by machine learning gradually improves with increased number of learning image. About 50,000 images were used to identify the endothelial cells. So many numbers of image we have to prepare, but after that, a machine can identify about 80% of accuracy. So we optimize many conditions. Here I change the input block size, difference block size, and the threshold. So this is a result, and this is a threshold, and this is a reference block, and this is accuracy marker. So as you can see here, lower threshold and higher threshold. Result is almost the same. Threshold difference didn't affect the accuracy. But in reference block size, larger image and smaller image. Larger image increase accuracy like this. So this data suggests a larger reference image size increase the accuracy of endothelial cell identification, which suggests that machine efficiently identify the endothelial cells by learning surrounding images of target cells. So we also optimize neural network condition. So we show here two networks, small network and large network. Large network slightly but significantly improves the accuracy like this. So network size also affects the accuracy of endothelial cell identification, which suggests that further network improvement would increase the accuracy. So next we optimize the wet and experimental condition. So as you can see here, this is specific signaling of endothelial cell marker. This is not specific signaling like this. So higher cellular density, edge of cell culture dish, but antibody quality increase non-specific signaling. Therefore, we have to eliminate those areas. Or we have to reduce the cell density to reduce those conditions. So we optimize in here and we eliminate those areas like this. After modification, network increase accuracy up to 95%. So image including non-specific signals decrease the accuracy. The elimination of those images or the optimization of cellular density increase the accuracy rate. So next we focus on where does machine learning shows the misprediction. So we focus on false positive area and false negative area. So prediction is stained, answer is stained. This is true positive. And so this is true negative. And this prediction is unstained, but answer is stained here. This is actually endothelial cells, but prediction is not endothelial cells. This is false positive, as you can see here. So cellular density is very important. Lower cellular density and higher cellular density, like this. So border zone of the cellular density and different cellular density leads to misprediction by machine learning. So in summary, we can identify endothelial cell from phase contrast image by machine learning. Accuracy rate is over 95%. Misprediction cases are heterogeneous cellular condition. In training stage, we prepared many phase contrast image, and then we also prepared answer set by immunostaining. And then we prepared endothelial cells and non-endothelial cells. This is the answer. So input into the network, and then network getting smarter. Then finally, they can identify this is endothelial cells, not endothelial cells, without immunostaining. So we developed a method to identify endothelial cells by machine learning technology. So we move on the next hypothesis. In same cells, but difference to status. I mean the same endothelial cells, but healthy and diseased cells. So machine can identify those cells. So we can develop the method to identify diseased cells. So we focus on aging. So aging is the strongest independent risk factor for cardiovascular disease. Many conditions induce aging: oxidative stress, inflammation, mitochondrial dysfunction, telomere attrition, epigenetic modification, genome instability, and dysregulated energy sensing, and impaired protein homeostasis. Those definitely induce aging, and aging induces cardiovascular dysfunction, and also cardiovascular dysfunction induces cardiovascular disease, such as hypertension, coronary disease, stroke, kidney disease, cognitive impairment, Alzheimer's disease, and motor disorders. So there is only one prevention strategy: lifestyle modification. But there is no pharmacological strategy. So to understand aging, so recently many studies conducted, and we found senescent cells have a central role in aging and aging-associated disease. This is aging cell, senescent cells. Senescent cells express several types of senescence-associated secretory phenotype, SASPs, cytokines, and hormones. So which induce aging phenotype, including immune disturbance, inflammation, the fibrosis, and tumor promotion, and tissue dysfunction. So we focus on several phenotypes of the young cells and senescent cells. In young cells, mitochondria is functioning, and so low protein oxidation, high proteasomal and lysosomal function. And after aging, senescent cells shows increased protein damage, enlarged nucleus, and damaged mitochondria. Lipoxygen is accumulated, reduced proteasomal and lysosomal function. So senescent cells shows unique features such as cell shape and intracellular structure. So if cell shape is changing during aging, machine learning technology could identify the senescent cells. So this is a strategy to identify the senescent cells and to develop the drug screening system for anti-senescent cells. We prepared the image of the healthy and senescent cells and put into the neural network. And then output is healthy or senescent. And then after development of the neural network, we use senescent cells and drug library. If a neural network answers this is healthy cells, probably this drug treats the senescent phenotype. So at first, we induce senescence in endothelial cells by several types of stress, H2O2, camptothecin, and replication. Camptothecin is an anti-cancer drug. So in this in senescence, it is well known. So this is a phase contrast image, and we cannot identify the phenotype. But actually, senescent marker, senescence associated beta-galactosidase activity is increased after stress. So beta-galactosidase positive cells is increased after stress, and senescent marker P21 and P53 is increased after stress. Then we developed the machine learning algorithms. We prepared many images like this. Then finally, we found accuracy rate is about 95%. ROC curve is shown here. AUC is 0.99. So this is good result of machine learning algorithm. Then we tried different conditions. So CNN was trained by either H2O2 or H2D in senescent cells. Then we examined this trained CNN H2O2 induced senescent cells. Trained CNN can identify replication stress senescent cells and computation-induced senescent cells. This is the result, and the classification accuracy was high in every data set. Importantly, the CNN trained by H2O2 induced senescence recognize senescence in CPT-induced senescence and replication-induced senescence. Similarly, the CNN trained by CPT-induced senescence and CPT and H2O2-induced senescence showed high performance over 0.95 in classification and under every condition. So these data suggest that CNN can reliably identify senescent cells, and the senescence is uniformly induced by any types of stresses. So it is often thought that machine learning is black box. We cannot identify what a machine identify. So here we use GradCam, gradient weight class activation mapping. GradCam uses the class specific gradient information into the final conversion layer of CNN to visualize the important region in the image. So this red area machine uses to identify the cell phenotype in healthy cells and senescent cells. In healthy cells, surrounding area of like this, smooth surrounding area is important to identify healthy cells. In senescent cells, intercellular heterogeneous organization is identified by the machine to identify senescent cells. So GradCam indicates healthy and senescent cells by recognizing various and heterogeneous intercellular image. This information could help us to understand the biological meaning of cell morphology. So let me examine the relationship between cellular senescence and the strength of senescence-induced stress. So we examine the several conditions of H2O2, 0.05 millimolar to 0.25 millimolar. As you can see here, P21 is increased by dose-dependent manner of H2O2 concentration. So this is the senescence probability output of the single cell level, one cell level. So this is a senescence probability. This is H2O2 concentration, higher concentration. Most cells show the senescence probability, and lower H2O2 concentration. Also, cells show the healthy cell. But actually, interestingly, there is huge intermediate state of cells, digitally transmitted from healthy cells to senescent cells like this. So we defined the senescence score to develop drug screening system. Senescence score is the overall average output probability calculated by the pre-trained CNN. So, and this is the senescence score. This is strength of the senescence induction, H2O2, CPT, and passage number. The senescence score is correlated with the strength of the senescence induction stress like this. So to validate the performance of senescence scoring system, the effects of anticellular senescence reagents such as nicotinamide mononucleotide, NMN, and metformin. So we examined SL beta activity. So metformin and NMN reduce their SL positive cells, and so senescence markers are also suppressed in those drugs. The senescence score is also significantly suppressed by metformin and NMN. So we move on the drug screening. This is a several image of healthy cells, senescent cells, and senescence with drug. So this is a result, and H2O2 induced senescent cells is here, and then we apply the library like this. And actually, most drug-induced senescence increase like this. So actually, most drugs are toxic. So we thought those most drugs induce senescence. But actually, some drugs suppress the senescence like this. So we examined several conditions. So H2O2 induced cellular senescence, replication stress induced senescence, and CPT, and then applied kinase inhibitor library. This is a heat map, and so this is bright color is the increased senescence. Dark color is reduced senescence, healthy cells. So we found this, there is an anti-senescence cluster like this. So anti-senescence cluster shows several drugs like this. Then we focus on 4 drugs among senescence clusters: taric acid, PD 98059, Diazine, Y27632. This is a MEK inhibitor, and this is a soybean extract, and this is ROCK inhibitor. So, and those drugs significantly suppress the aging phenotype. So to understand the mechanism, we added those drugs in endothelial cells and performed RNA sequence analysis. And we focus on differentially expressed genes, and we applied gene set enriched analysis, and we found this drug reduce the NF-kappa B inflammatory response mediated gene expression is suppressed by those compounds. And among them, we also focus on taric acid because there is no report about the taric acid and aging. So we focus on taric acid and we performed RNA sequence analysis. And we found taric acid improves mitochondrial function, ATPase activity, and oxidative phosphorylation. So in conclusion, we can develop the system to identify endothelial cell senescence like this. And then those systems, we also developed the drug screening system. And then we found several anti-senescent drugs. So machine learning can identify the cellular status without molecular technique. Accuracy rate is approximately 95%. Machine learning-based drug screening system could identify potential drugs for aging. Finally, I'd like to thank for many collaborators in our laboratory and also Sony company. Thank you.
Okay, thank you very much for your very nice presentation. You had a very beautiful data here. So yes, thank you very much. I was surprised that currently you are interested also in the senescent study and also doing the drug screening for the senescent endothelial cells. Maybe as you know that we had many herbal medicine here as a jamu, and maybe in the next time or in the next collaboration research, we can screen out the herbal medicine in Indonesia for the anti-aging drug in the senescent. And also, I believe that many participants here also has many experience for the stem cell also, but and maybe some of you will ask some questions to the Yuasa sensei, but please keep the questions and we will proceed into the second speaker here. So for the second speaker, so we will have Dr. Wawaimuli Arozal. So she will give a lecture about the recent clinical relevance and translational limitation of animal models in cardiovascular medicines. So as we know that Dr. Wawaimuli Arozal is a head of Department of the Pharmacological and Therapeutics in Faculty of Medicine, Universitas Indonesia, and also the associate professor in the Universitas Indonesia. So she has achieved her M.D. and Master in the Universitas Indonesia and also got the Ph.D. in the Niigata University. And currently she has many publications One of them is in the frontiers of cardiovascular medicine as a first authors this year. Congratulations, Dr. Wawaimuli. And also during her research, she had developed the cardiovascular disease model in animal models, such as the doxo or the neurobisin induced toxicity in the cardiovascular, the autoimmune myocarditis, the diabetic cardiomyopathy, and also the swimming test for the cardiovascular analysis. So it must be very interesting to hear about her excellent experience in this presentation, particularly for making the cardiovascular disease in animal models. So please, if you are ready, Dr. Wawaimuli, you can start the presentation. Thank you very much.
Okay, thank you very much. If voice is clear, Dr. JP?
Yes, yes, please.
Thank you. Thank you very much, Dr. JP, for the nice introduction. So good afternoon, all participants of doctoral program webinar series. I wish all of us is good, healthy in this pandemic situation. So good afternoon also, Yuasa Sensei. Thank you for your interesting presentation. There is a very advanced technique for cardiovascular research. Before I start my presentation, allow me to say my gratitude towards the organizer of this event, who has given me the opportunity to share my lecture on today's webinar. So in this webinar, allow me to present my lecture regarding the recent clinical relevance and translational limitation on animal models in cardiovascular medicine. So my presentations for this day will be divided into several sections. So especially regarding various stage of cardiovascular drug development. And then I also will focus on the several in vivo models which are commonly used in as an animal model on cardiovascular disease, alongside its limitation and the relevance for clinical application. So as already Yuasa Sensei also mentioned about the cardiovascular disease is still a leading global cause of morbidity and mortality, similarly also in Indonesia. So about 31% of all death in the worldwide, I mean in 2011, I don't know the exact frequency nowadays. So additionally, 34% of CVD attributed deaths occurred before 75 years of age. So in we found that in Indonesia also similarly that now young people commonly dead because of the CVD problems. So according to the WHO organization, some of the top cardiovascular related causes of premature death, including the coronary heart disease, chronic obstructive pulmonary disease, and also the stroke. So this figure is show you that chronic CVD is still the leading cause of mortality in the world, followed by cancer, respiratory disease, and also digestive disease. On the other hand, the mortality caused by infection is lower compared with the chronic disease. But if we take a look at this figure, so the development of CV drug in this line, in the orange line, so it tends to slow and tend to plateau compared with the oncologic drugs. Okay, so because of currently oncologic drug development is remarkably fast due to the increase in the development of life-saving targeted therapy. So there are many developed drug related to targeted therapy in oncologic field. So the other problems which cause that the delay of the CV drug development may be because of the bad marketing or the lack of breakthrough in drug development in the CV disease, CV treatment. So as I know that in Indonesia, so one of the breakthrough CV drugs is ARNI class, angiotensin receptor neprilysin inhibitor, that it is approved by our FDA last year, yeah, if I'm not mistaken. So the ARNI class is for the heart failure with reducing ejection fraction. Okay, so this figure show the process of drug development, starting from the lead discovery until clinical study in human. So as we can see also, in vivo study in animal is used to elucidate the mechanism of action, and then behavioral studies, and then also safety and efficacy before it is go to the clinical trial. Okay. Yeah, so in this figure, we can see that how a drug development process requires a very, very long time, yeah, from lab to market. From the 1,000 of substance, yeah, about 5,000 or 10,000 compounds, it will be reduced to 25 compound in preclinical setting, and then only about 5 compound can be go to the clinical trials, and then only very less can get the approval from the regulatory. So it means that we need to faster, yeah, to have any breakthrough, yeah, related to the drug development. Yeah, so it means it is need a lot of money, yeah, to get, I mean, to a lot of funding for research to find one drug start from the drug discovery until regulatory approval. Okay, so one of the animal model used especially for screening is well-known as zebrafish model. So on several countries, zebrafish is not categorized as an animal model. So it is not categorized as animal. So the zebrafish model has been used for decades to study development and pathophysiology. So more than ever, the specific strength of the zebrafish model make it a prime partner in the age of discovery transformed by big data approach to genomic and disease. So zebrafish also share a largely conserved physiology and anatomy with mammals, and then also allow a wide range of genetic manipulation, so including the latest genome engineering approach. And then the zebrafish also can be bred and studied with remarkable speed, enabling a range of large-scale phenotype screen. So in this figure, we can see that several advantages of using a zebrafish model, such as the easy to genetic manipulation, and then it is easy to study the anatomy and physiology of the zebrafish. So in zebrafish also we can easily take a look by imaging. So the whole organ as well as also the subcellular resolution imaging can be seen in this kind of model. Okay, so several animals model are applicable for zebrafish, especially animal model relating to CV and metabolic disease, as can be seen on this figure. So in Indonesia, I mean, one of our institution in Bogor, so they developed the screening model using zebrafish, yeah, for candidate for diabetic mellitus treatment. So they check many compound, yeah, herbal compound, and then they screen it using the zebrafish model. So it is easy to make the fish become hyperglycemic, yeah, and then distract the beta cell pancreas. So, but this kind of model is not to prove the pharmacodynamic or to prove the efficacy of some certain drug, but only for screening method. Okay, so Okay, so these figures show the several advantages and disadvantages using animal model, especially for study in cardiovascular area. So, for example, in animal model, we can control everything. So, control like the food, the situation, the temperature, everything. So, and then also we can check many kind of different model in animal. But the limitation of the animal models is because of there is a difference between human and animal, even though it is a mammalian. Yeah. So they also anatomical difference, and then the pathophysiology mechanism may be different between animal and human, and then we have also to consider about the result in vitro versus in vivo. Maybe there is some differentiation, something like that. And then some drugs also the response is maybe different. Yeah, in the same group of drug, maybe the response is can be different. Okay, so one method to study CVD using animal model is through genetic manipulation of the animal, such as a mouse or a rat, to produce an animal model with an appropriate CVD without surgical or chemical induction. So maybe all of the students familiar with the knockout model, but unfortunately in Indonesia we cannot breed this kind of animal. Yeah. So because of our, the lack facility of our animal house, something like that. So the procedure also need a specific treatment, and then also the specific instrument for adequate breeding facility. Okay, so here I will show you some animal models of atherosclerosis. Yeah. It can be done in mouse, yeah, but there are some advantage as well as also limitation to do atherosclerosis in mouse, yeah. So in Indonesia, in our institution, we are commonly using mouse to induce with high fat, high cholesterol diet, yeah, to induce the atherosclerosis disease. And also we also can do the atherosclerosis in rat model, yeah. But our BPOM, FDA regulation, they suggest us to do the atherosclerosis model in the rabbit, yeah, because of in rabbit, the high level of LDL as well as also the high level of cholesterol is not reversible, yeah. Irreversible. In the rat, sometimes after 2-3 weeks, yeah, after we induce the rat with a high cholesterol, the LDL, the cholesterol level will be tend to the normal. But in the rabbit, so it's still in the high level, something like that. Okay. But also there is some limitation in each animal, yeah. And also we can do in porcine, for example, but of course it is high cost and then difficult to handling, and then a few genomic tools. Okay, and then we also can create the animal models for plaque rupture and plaque associated with thrombosis. For example, if we use by spontaneous rat, yeah, using genetic mice as well as also genetic rat, yeah. Unfortunately, again, we cannot develop this kind of model because of the lack of facility, yeah. So and then we also combine genetic mice and then genetic rat with some kind of induction, yeah. For example, using balloon injury or some high cholesterol diet, or in combination with angiotensin 2, so to induce the rupture of the thrombosis. Okay, and then we also can create aortic aneurysm, yeah, by using some animals, yeah. For example, in murine or in mouse, yeah, we can induce the aorta to dilate the aorta by injection with calcium chloride or using elastase infusion, or we can induce with angiotensin II and many kind of technique, yeah. Also using xenograph, yeah. And then we also can induce aneurysm in the rabbit, in the pig, yeah, using surgical model. Yeah. So now I would like to take a look with the animals models in heart failure. So in our institution, we create many kind of model for heart failure because of, you know, that the incidence of heart failure in our hospital, I mean in Indonesia also every year is tend to increase, yeah. So we hope also our natural product, yeah, maybe have beneficial to reduce the progression of heart failure. So in the murine model, in the mouse, so we can induce the myocardial damage by a chemical, for example, like isoproterenol. So I'm frequently doing this kind of model, yeah. So as you know that isoproterenol is a kind of beta-2 agonist. So in the in the therapeutic dose, it will increase the contraction, but in the high dose, it will induce the necrosis of the myocyte itself. So it is, I think it is easy to do this kind of animal model, yeah. Okay, and the others model is, for example, like surgical coronary artery ligation. Maybe Dr. Jeffy familiar with this kind of model, yeah. And then the others is using pressure overload, bending aortic, yeah. So it makes the aortic stenosis, yeah. And then some transgenic model, for example, we can do the modified the cardiac alpha actin protein. So it means the myocyte will be damaged. Okay. And then for the others, animal can be used for heart failure, for example, rabbit and then dog. Yeah, maybe in some country they are familiar with this kind of animal model using ventricular pacing. But again, maybe there are some limitation, maybe related to ethical issue. Yeah, if we using large animal. Okay, some animal models, including mice, rats, and guinea pigs, continue to improve the understanding of the various aspects and etiologies of heart failure and help to develop novel treatment strategies. Yeah. Mice and rats, especially also in our institution, is the most commonly used animal models and share a high degree of homology to the human genome. So major advantage of rodent models include a relatively short breeding cycles and low housing costs. Yeah, so this figure show you the pathophysiology of heart failure. So as we already know, the heart failure clinical symptom is clinical signs is divided into two, yeah. Heart failure with reduced ejection fraction and then heart failure with preserved ejection fraction. So so I will not teach you about the pathophysiology, but we know that the hypertension and then obesity, yeah, and then also myocardial infarction is the most common of heart failure, yeah, the etiology of heart failure, also in Indonesia. Okay, so I would like to, I would like to mention about how we can do the model for reduced ejection fraction in small animal models. So we can do in many kind of technique, yeah. For example, in aortic constriction. So and then using the ligation of the artery coronaria. Yeah. So and then we can do also the pulmonary artery constriction, fistula of aorta cava. Yeah. This technique is need the the expertise to do the surgical process. Yeah. So we also have to make sure that our lab is very sterilized. Yeah. So we have to avoid the infection. Okay. So the model will be good. Yeah. So the easy way to do induced heart failure is using the chemical. Yeah. So for example, like doxorubicin and then isoproterenol. But the pathophysiology or the pathomechanism is slightly different with the clinical setting in human. Okay, so I searched in one article. Yeah, it is recently published. They introduced the new animal model for heart failure with preserved ejection fraction. Yeah, so the mice induced with the high fat diet, and then also induced with the L-name. Yeah, L-name, and then it will develop the obesity and then glucose intolerance and also high blood pressure. As you know that L-name also induce the hypertension in this animal model. So the pathomechanism how L-name can develop the heart failure in preserved ejection fraction is by the reduce the activation of this kind of expression, yeah, IRA1 alpha. So the translation of XBP will not go through, and then it will decrease because of the less activity of this kind of gene. So it will further develop the impaired cardiac feeling and then inability to exercise, lung congestion, and then also inflammation. So I think we can try this kind of model. Okay, so this is the summary of how advantage as well as also disadvantage using large animal model. So large animal model means dog and then pig, and then also some research they use monkey, yeah, to do the clinical to do basic research. So I already mentioned about the problem, also the cost, and then also the ethical issue. Yeah. So some of when we are submit our method in the ethical ethical clearance, so sometimes they will not allow us to use the large animal model. Okay, so this also will show you the positive and negative of using small animal models and large animal model. So for example, like in large animal model, it is closer, yeah, to the human anatomy compared with the small animal model. Yeah, so better tissue availability, and then so if we want to check also the pharmacokinetic profile, for example, it is better to do in large animal model compared with the small animal model. Okay, so this figure show you that some large animal species, for example, like pig, yeah, it can be induced for a pressure overload model, yeah, and then dog also can be performed for myocardial infection, yeah, using ischemia, reperfusion, or permanent coronary occlusion or coronary microembolism. So it means that we have to has expert, yeah, in surgical, yeah, in surgical. Okay. Yeah. So this one also some summary related to the large animal model. Some large animal can be done for preserve ejection fraction and also for reduce ejection fraction. So many kinds of model we can done. Yeah. Also we can done by using glucocorticoid, yeah, to induce the hypertension, yeah, and then after that it will be develop the heart failure. So I think this kind of phenomenon also Dr. Yuasa already mentioned related how we faster, yeah, the process, yeah. So in this figure we can see that due to the limitation of using animal model, data from human sample can be used, yeah, that data from a human sample can be used as a starting point in determining targeted gene for therapy, which will be further tested in silico to find a substance which correspondent to the targeted gene. The next step then to test it to identify the phytomechanism and then test to the toxicity in the animal, yeah, before finally moving to long-term study and clinical study in human. So just we can, try this kind of step, yeah. So from, for example, if we take out a cell, yeah, as Dr. Yuasa mentioned before, yeah, maybe we can check the endothelial cell from after surgery, for example, and then we can find, targeted gene, yeah, something like that, and then we start from there. Okay. Okay, so I'm sorry if not clearly seen, yeah, but in this table, I would like to share you the different anatomy, yeah, the selection, similarity and difference between human and animals, yeah. Start from zebrafish, mouse, rat, yeah. For example, like in rat, the heart rate is faster than the human, okay. And then the systolic blood pressure and also diastolic blood pressure is higher than in human. Yeah, so you have to consider about that. Okay, so the coronary artery anatomy also slightly different between each animal. Okay, so now I'm coming to the conclusion. So as I already mentioned, there are no ideal animal model, yeah, for each cardiovascular disease. Yeah. A model is just a model. So the cardiovascular system of each animal has evolved differently in order to meet the demands of that species. Animal research is lacking the possibility of true long-term follow-up. Yeah, this is the problem. Okay, readily seen and appreciated in clinical trials. So that's why in basic research we find about more than 200 substance. It is promising, yeah, but while studying in human, the efficacy is lack, something like that. Okay, so we have also to consider about the biologic cause and then including confinement, pain, suffering, and death of the animal and most patient. And then no ideal animal model of the human cardiovascular system exists, and studies should not rely on a single animal model. So I think it's better to us to do in many animal model, yeah, to address all questions. And then given the complex nature of cardiovascular disease, no species or animal model will present the exact simulation of the human disease. Therefore, models should be chosen according to the study aim. And then the ideal animal model of CV disease should mimic the human subjects metabolically, as well as also pathophysiologically, be large enough to permit physiological and metabolic studies, and develop end-stage disease comparable to that in human. So as Dr. Yuasa also mentioned about what should be in the future, yeah. So animal research needs to evolve for single to multi-disease model to avoid future pitfalls and translational failure when advancing from preclinical animal models into the human setting. And then we have to consider about the humanize the mice and then genetically modified a large animal model. So in the future, maybe we can create the artificial intelligence base as to develop the new candidate for drug. So this is my reference. And then thank you very much for your attention. So I'm very happy to answer the question if any.
Okay, thank you very much, Dr. Wawaimuli, for the very interesting and comprehensive lecture. So we have several questions here. Okay, so let me start for the discussion sessions. So from the Miss Bantari, her question is, do you mind to share how to solve the misprediction cases in identifying endothelial cells using machine learnings? So actually, Dr. Yuasa already answered here. We found that the homogeneous cellular condition is very important. Therefore, we reduce the cellular density. So is there any additional comments, Mrs. Bantari or maybe Dr. Yuasa? So please, if you want to give additional comments, thank you.
So we analyzed the misprediction case and we found an both zone of the heterogeneous cellular density and the several conditions affect the cellular density. So we carefully cultures to be homogeneous cellular condition. Also to match cells in this heterogeneous cellular condition. So we reduce the cell number. So wet condition is highly important.
Okay, thank you very much, Dr. Yuasa. So is that answer your questions, Ms. Bantari, or we can proceed into the next questions? Is it fine? Or yes? Oh, okay, it's fine. Thank you. So we can go to the next questions from Dr. Dewi Sukmawati to Yuasa Sensei. The first question is, what are the basic components to develop the research using AI methods? And the second question, what kind of experiments that be suitable for the AI method, especially in the cardiovascular field? Thank you very much. So please, Dr. Yuasa.
I cannot understand what means a component. What kind of component do you mean?
So, Dr. Dewi, do you have any additional comments? You can chat or maybe you can directly talking.
Okay, thank you. So maybe component is like, okay, you mentioned about the big data, maybe about the materials or something else that we should prepare in order to use the AI method.
Okay.
Any additional materials or other than the big data? Is there any other?
You mean the computer, kind of computer and server?
Yes.
And also, we of course we have to use a little bit high ability computer and also server, and so we have to write a program, computer program, but it is not so difficult. So,
so I mean, we have to develop the program first, and then I mean, we do the experiment as usual, and then plus the program for the AI method. Is that correct?
Yeah, but actually in AI technology, it is very important to accumulate good data. So many data, and so its quality is also very important. So in our case, about 100,000 images we collected. So many images is needed.
I see. Okay.
Okay, is that answer your question, Dr. Dewi? Or you have any additional comments?
Yeah, for the first question, I think it's fine. Maybe the next question, the second question.
Okay, Dr. Yuasa. So the next question is, what kind of experiments that be suitable for the AI methods, especially in the cardiovascular field?
Well, whatever you want, we can apply the AI, but actually, as I mentioned, we have to collect many data. So in the field of research, if we can get the big data, we can apply AI method. Depends on the data, I think.
So I mean, beside the image, for example, can we measure something like concentration or other things?
Whatever, for example, ECG, X-ray, anything is okay. RNA data, it's okay. But we have to get many data. That's that.
Okay, thank you.
Okay, is that answer your question, Dr.
Yes.
Dewi?
Yes.
Okay,
Yeah,
thank you very much, Dr. Dewi and Dr. Yuasa. Okay, so we can go to the next question from Dr. Puspita Katili. So she has a second question, two questions. The first question is for Dr. Yuasa. So, Dr. Yuasa, is this machine learning has similar principle with the path reader such as Opereta? As Opereta also have a software that we can train to detect such as an alignment, whether chaotic or organized, and background intensity, so it can differentiate the specified population. Also, is this suitable for 3D platforms such as the engineered heart tissue? Oh, it's very advanced question. So please, Dr. Yuasa.
Actually, I don't know the operator, but probably it is similar protocol. So probably it is commercially available. So if you use that for your purpose, it is better to modify the program to fit your research. So it is better to use and generate your own program, but you can use probably operator for your research. So
okay.
Another question is 3D platform. This is also very interesting, but as I mentioned, it is very important to get many data. So 2D flat cultured cells, it is easy to obtain many data. But 3D culture condition, so probably data quality is decreased. Therefore, much more data is needed.
Okay, thank you very much, Dr. Yuasa. So, Dr. Puspita, is that satisfy your questions? Okay, thank you very much, Dr. Yuasa. And for the next question, for the Dr. Wawaimuli from the Dr. Puspita. So, for cardiomyocyte research using small animal model, rapid has been proved to represent human model more compared to mouse due to the positive staircase effect in contractility. For example, increased force with increased frequency rapid, which is the case for human compared to negative staircase effect of a mouse. Will UI means maybe Universitas Indonesia use rapid in the future research instead of mouse? Dr. Wawaimuli, please.
Yeah, thank you very much for your question. So I agree with you. Yeah, I mean, in certain animal model, for example, like atherosclerosis model, in my opinion, it is better using rabbit instead of using mice or using rat, because of the level of the cholesterol, so we can maintain the high level of cholesterol if we using the rabbit instead of the small animal model. So it can be easily to manipulate the rabbit by food to induce the atherosclerosis. And then, for example, we can easily check the aorta abdominalis. So if using rat or using mice, it is difficult to find out the pathology of the aorta itself. So we are also thinking about that. So in our animal research facility in IMERI, maybe Yuasa Sensei, you already come to our animal research facility or not last year? in IMERI. Okay, so in our animal research facility also we have a specific room for rabbit. So maybe in next next research, our group, Dr. Jeffy maybe will perform animal model in rabbit to develop some CV disease. I hope it is answering your question.
Okay, thank you very much, Dr. Wawaimuli. So as a, if I might to give an additional comment, so as a Dr. Wawaimuli's in the presentation, in her presentation, so she talked about the Watanabe rabbit. So this Watanabe rabbit actually was developed in my previous college in the Kobe University and has been used for the LDL receptor invention and awarded a Nobel Prize. So maybe I don't know if we can transfer this rabbit into Indonesia.
Yeah, okay.
So
maybe
yes.
Not easy to breeding that kind of rabbit. Yeah.
Yeah. Oh, maybe thank you very much, Dr. Wawaimuli. So, Dr. Yuasa, maybe you have some additional comment for using rabbit, maybe in the IPS and cardio study, maybe.
Mm-hmm. Yeah, many model is important to understand the mechanisms to develop the therapy. So depending on the purpose, we have to choose the models. Mouse and sometimes very important to generate genetic model. And so big animals like pig and also sheep and monkey. Sometimes we use for some purpose. And the IPS cells is also very important and also useful to understand the genetic disease, human disease. So depending on the purpose, we choose the models.
Okay, thank you very much, Dr. Yuasa. So, okay, we can go to the next question. We have Dr. Jeffman here. So, Dr. Jeffman would like to ask directly to Dr. Yuasa. So, please, Dr. Jeffman.
Okay, thank you, Dr. Jeffy. I would like to ask to Yuasa Sensei. My first question is, in the term of identify endothelial cells, you need a huge or large data to make a detection endothelial. How about the developing a new drug, a potential substance to become a new drug? What data do you need to generate AI approach to identify this potential new drug? That's a first question. The second question, do you have any experience maybe using the AI approach in a clinical research setting? If you have, maybe you can search with us. Thank you very much.
Okay, okay, please.
Mm-hmm. And the first question is, so drug development. So firstly, it is very important to collect the data, big data, to develop the first network to identify diseased cell or not diseased cells, and endothelial cells are not endothelial cells. After establishment of the network, we don't need so much data. So drug screening, we don't have to accumulate the same number of data. The initial development is very important. So after that, we don't need that. And the second question, clinical data. So we also applied those methods to clinical research, but actually it is very difficult to collect the very rich number of data. So X-ray data, ECG data, so very difficult to obtain the many data. We tried, but it is very difficult.
Okay. Do you have any additional comments, Dr. Jeffman?
No, Dr. Jeffy, thank you, Yuasa Sensei.
Okay, thank you very much. So maybe if it's possible, I would like to ask another question related with the Dr. Jeffman, Dr. Yuasa. So actually, in your I study in senescent endothelial cells. So you can simply
identify the senescent just using the II without using like any staining such as beta gal staining, just by the morphology itself. Is it right? Okay, because as you know, maybe the morphology of the senescent cell is a diverse, like a flattening, irregular pattern. Yeah, it's.
So at first we also think if we apply the stress to the cells in cell culture dish, probably, and for example, 7% is senescent cells and 30% is healthy cell. So I'm afraid it is difficult to identify. But actually, after we tried, probably small differences already 100% cells. So machine can identify the every cells. So in same cell culture dish. So as you're afraid, diversity is in human eye, but probably in computer, in machine can identify every cells.
Yeah, very interesting. And do you think that is it for the analysis itself, is it possible to can we use here in Indonesia in our faculty? I mean, did you need some sophisticated software or some advanced PC for developing the II for detecting the senescent cells?
Actually, we don't need a high-spec computer. It's low-spec computer, but to efficiently perform AI technology, we have to develop sophisticated program. So try and error. We have to try many times to develop better algorithm. So you can do it, but it takes a little bit trials.
Okay, so yeah, very interesting. So I'm very eager to read your paper in the new Nature Communications, and maybe we can do the future collaboration research regarding this issue. Okay, thank you very much, Dr. Yuasa. And we can go to the next question from the Ms. Bantari again to the Dr. Wawaimuli Arozal. So cardiovascular disease research as well as metabolic research is sometimes hard to conduct successfully in the in vivo models. Do you have any tricks or any suggestion for the preferred method? So please, Dr. Wawaimuli.
Yeah. Okay, thank you. Very nice question. So I agree with you. Yeah, it is sometimes very hard to conduct successfully in vivo model. Yeah, so to develop the cardiovascular and then also for metabolic. Yeah, so because of the metabolic mechanism is from animal and human is different. Yeah, itu JP. Yeah, so not exactly similar. So. So I suggest to do many preliminary study, yeah, to to achieve the pathology condition, yeah, doing preliminary study, and then maybe you can perform not only one animal model, so you at least you perform 2 or 3 animal models, so so you can get the validity data, something like that.
Okay.
So we have to try, of course.
Yeah, of course. Okay, thank you very much, Dr. Wawaimuli. So, is that answer your question, Ms. Bantari? Okay. Okay, thank you very much. Okay. Okay, for the next question, we have from Ms. Leonie Dwirskita. So it's very interesting question actually. So regarding to your preference in making heart failure model for in vivo, and do you mind to tell us why you choose the method? So as we know that you already presented the heart failure preserve ejection fraction using the LNM and also high-fat diet. I think that's a very interesting study. So please, Dr. Wawaimuli.
Sorry, actually, I also find this kind of new model in new published paper. Yeah. So they introduce the new model. So I think it is likely easy, yeah, to develop because of we know how to give to treat, yeah, to induce a high fat diet, yeah, and then LNM also not too much difficult, yeah, to find. So I think it will be 100% will develop the heart failure with a preserved ejection fraction. So I think we can try this kind of model because of it also need only about 8 weeks, yeah, if I'm not mistaken, to develop the heart failure. So you can manipulate and then you can also treat many kind of treatment. So the reproducibility is very good, I think.
Yes, yes.
Okay, thank you.
Yes, thank you very much, Dr. Wawaimuli. Yeah, I think we can translate or apply this method in Indonesia since it's not so difficult to apply. So is that answer your question, Dr. Leonie? Okay. Okay, so we have maybe some additional question from Ms. Leonie. Can we have the paper that you mentioned? Or maybe, yeah.
Yeah, so
you can share the link maybe,
yeah,
yes. Okay,
we
thank
will
you very
send
much.
the link. Yeah.
Okay, okay. So we have a next question from Dr. Denny Rahmat for Dr. Wawaimuli. To choose the right animal for the experiments that we can apply for human, do we need information about the type of drug or maybe their biodistribution in our animal? So please, Dr. Wawaimuli.
Okay, oh yeah, of course lah, yeah, we have to also consider about the, I mean, what kind of drug administration we have to choose, either it is intraperitoneally or orally, and also maybe intravenously, something like that. So, I mean, but in our regulation, yeah, in BPOM regulation, yeah, to perform animal models, yeah, to do registration process. So the route of administration it should be same. So for example, we will give the patient using orally. So start from the animal model should be by using orally, something like that. So we have to know exactly the kinetic profile of this kind of substance. I agree with you. Yeah, thank you very much.
Okay. Is that answer the question, Dr. Denny Rahman? Or you have any additional comments?
Yeah, okay.
Okay,
the
thank you very much.
Biodistribution also, maybe we have to know about that. Yeah, the distribution of the, for example, if our target is in the cancer cell, for example, like, our target is in the joint, something, if possible that that kind of drug is can be distributed to the joint, something like that, we have to know the I mean the kinetic profile of this kind of compound.
Okay. 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 Yeah. Again, thank you very much, Dr. Wawaimuli. Again, we have another question for Dr. Wawaimuli. So maybe I saw here some of the expertise in the stem cell already joined this meeting, such as Dr. Radiana, Dr. Dewi, Dr. Norma, and Dr. Puspita, and any else. So maybe you can ask the question to Dr. Yuasa. From Dr. Melva Luisa to Dr. Wawaimuli. Oh, okay. So this is just a comment. Dr. Wawaimuli conducted many studies in cardiovascular model. Dr. Leonie can search her studies in PubMed or Scopus database. Okay, thank you very much for the information. Okay, so we have also Prof. Asmarina and maybe Dr. Nafialdi here. Thank you very much. So if you have some questions for Dr. Yuasa and also Dr. Wawaimuli, so please you can ask the question here.
Yeah, I would like to ask to Professor Yuasa. Thank you. Yeah, I cannot, I could not join as your presentation today because I have examination for my student. Yeah, it's pity. But yeah, I would ask about the general question actually about the AI. Yeah? If we use the AI, yeah, we have to develop a software. Yeah, it is important to collect the data, the big data, and so on. Have you already developed by yourself or by your team in this software? And then yeah, I think as we as a faculty, yeah, in our university, yeah, how we can, I mean, yeah, the software is not cheap. Yeah, I mean, there is it is needed so many knowledge from the computer science and so on, and combined with the medical expertise also. And then the software that has already developed, I think, is suitable only for this lab. I mean, can we in Indonesian, for example, can use only this software if you have, for example, if you have already developed, or it is better if we develop by self for this software, or yeah, it's depend on the software that we develop to evaluate all many diseases. I mean, so how? Yeah, the second question: how can you develop this software, and then can we also use only your software, for example, and then we can purchase the software or something like that to our faculty?
Thank you your comments. And also, actually, we develop the software by our laboratory, by our own. Actually, young researcher, most young researcher, even in your facility, probably can do it. Not so difficult for young researcher. So just push them to develop something. Also, in each project, in each purpose, software has to be changed. So purpose. So always developing is needed. So we don't have to purchase, just develop. So
okay. And then if we want to use your software, for example, must we purchase this software or something like that? Yeah.
No problem.
Okay.
Anyone can use it. But if we want to do something new, we have to develop next new software. Always write the computer program.
Okay. Okay. Okay, maybe in the near future, if clinical scientist or clinician will also use this platform or use the application, yeah, to diagnose the disease, yeah, in cardiology. I think maybe we can use also the software, yeah. Maybe it's collaboration with your lab, maybe to use this in our university. Or so maybe. Yeah. Thank you, Dr. Yuasa.
Thank you.
Professor Yuasa. Thank you.
Okay. Thank you very much, Prof. Asmarina, and also Dr. Yuasa. Okay. We still have time, maybe the remaining time, the 15 minutes, is that right? So we can still have any questions for these sessions, maybe. If someone wants to ask the question, you can ask here directly or maybe through the chatting. Okay. Okay, anyone? Or maybe can I ask the question to Dr. Yuasa? So this is the naive question actually. So actually we have some expertise here for the stem cell research, and also the facility, of course. So for the big question, what is your recommendation for developing the IPS laboratory in our faculty? I mean, just a brief answer or brief explanation, not so specific explanation. So do you have any suggestion or recommendation? Thank you, Dr. Yuasa. Yes.
Okay. So human iPS cell is generation of human iPS cells is not so difficult. But so most important is a little bit cost. So culture media is a little bit expensive. So long-term culture, it is very cost necessity. Therefore, if you have some project, so just focus on the short-term project is important. So for 1 year, 2 year, 3 year project is some very, very expensive. So just 1 month and 2 months is better for you to consider the project.
Okay.
iPS generation, differentiation, and is well established. Not so difficult, but cost is very important.
Yes, the cost. Yeah. Thank you very much for the answer, Dr. Yuasa. Okay, so we have Dr. Jeffman again. He would like to ask the question. So please, Dr. Jeffman.
Thank you, Dr. Jeffy. Yuasa Sensei, I'm a little bit curious about your software. I mean, do you use specific software, or you use R, Python, or RapidMiner maybe? And I mean, do you develop a software, or do you develop an algorithm to make an AI approach? So that's make me a little bit curious about the software you use. I mean, which one is preferred if you use Python or R? Thank you.
Both of them we used Python and R. So whatever you want, it's okay. Sometimes C language is also sometimes very important, but whatever you want and whatever you likes, you can choose to do that. The Python is commonly used, I think.
And I'm sorry, do you have any specific model you use to develop the algorithm maybe?
Not so specific. Usually, image recognition system is CNN, convolution neural network, used. So we also use that. And some program should be modified, just modification. Basic algorithm is already established.
I see. Thank you very much.
Okay. Thank you very much, Dr. Yuasa, and also Dr. Jeffman. So, okay, is there another question for Dr. Yuasa or also for the Dr. Wawaimuli? Not only for the artificial intelligence, maybe related with the iPS cardiovasculars and maybe the cardiovascular models in animal models. So please. Okay. So actually, I also have some issue for the iPS cells for Dr. Yuasa. So as you know that the iPS cells can be collected from the fibroblasts from the patients, but maybe in Indonesia we have some ethical issue for collecting the fibroblasts from the patients. So I read another paper explain that we can make iPS cell from the blood cells, the peripheral blood cell. Do you have some comment about it? And is it possible to perform the iPS cell from the blood cells?
So now it is widely accepted to use blood cells because less invasive for the patient. Around 2008, we also obtained the human dermal tissue. It is very painful. So we developed the method to generate iPS from blood. So it is better to use blood, but actually in January, we use Sendai virus to generate iPS cell from blood. So we have to buy Sendai virus. But in fibroblasts, we use retrovirus. Retrovirus is free. So less cost effectiveness is good for the fibroblast. But for the patient, it is not so good.
Okay, so is it commercially available for the Sendai virus?
Oh, yeah.
Okay. Okay, thank you very much. Yeah, maybe next time we can invite you to our faculty to supervise us, maybe after the pandemic is over, maybe. Thank you very much.
Thank you.
Okay, is there any another questions? Okay. Okay, for the Dr. Wawaimuli, maybe if I can ask the question. So, as you mentioned, there are several limitations for doing some research in the cardiovascular research in animal model. So, do you have any suggestion that maybe is there any model for the cardiovascular model that we can do or we can apply in Indonesia maybe?
So for which indication? For what kind of indication? For example, maybe in my expertise, so I many study related to heart failure model. So previously when I'm in doing my PhD work, so I'm study related to doxorubicin induced heart failure. So it is easy to induce. To induce the heart failure using the doxorubicin. So only we need 2 weeks, 2 weeks injection using doxorubicin, and then after 2 weeks we can get that the contractility of the heart become reduced, something like that. So. And then also the mortality rate. Yeah, we can perform. We can treat the rat until 8 weeks. It means the mortality rate after injection with doxorubicin is not so much. Yeah. So if we, in the different chemical, for example, isoproterenol, yeah, the mortality rate is very high. So, for example, if we inject it with 10 rats, so only 5 rats will be survived for 8 weeks, something like that. Yeah. So we have again to do preliminary research. So which one is your convenient for that? Okay. So actually the strength of the rats also will different. Yeah. For example, Wistar rat and then Sprague-Dawley rat. For doxorubicin induced, I suggest you to do with Sprague-Dawley rats rather than Wistar rat. So you know that in Indonesia there are only 2 kinds of species. Yeah, we can order only SD as well as only Wistar. The other species cannot be purchased in Indonesia. Thank you.
Okay, thank you very much, Dr. Wawaimuli. So maybe Dr. Yuasa, do you have any suggestion maybe for making the possible cardiovascular model in the easy way or the simple way, but have a high impact for the cardiovascular disease that can be we apply here in Indonesia, maybe, Dr. Yuasa?
Actually, we often use just TAC model.
Okay.
It's a respiratory physiological hypertrophy, therefore enough heart failure. So generally we use that. Also, it is better to use knockout mice. So ideally, conditional knockout mice is better to understand the disease mechanism.
Okay, so and also the recent paper that Dr. Wawaimuli previously presented from the Nature that they use the L9 and also high fat diet for high heart failure preserved ejection fractions. So have you read or do you have some comments for it? Is it possible?
I didn't read it.
Okay. Okay, thank you very much, Dr. Yuasa. Okay. Okay, so is there any additional question for Dr. Yuasa or maybe for the Dr. Wawaimuli? Okay.
Dr.
Yes.
Jeffy,
Yes,
yeah.
please.
May I
Of course.
have one question. Okay, for Dr. Yuasa, can you suggest us which cell from the rat or mice that we can use here and we can develop for the IPS model? I mean, the easier maybe because we still have some limitation here.
There are many data about the mouse, but rat is very rare, and it is difficult to generate stem cell from rat. It is possible but difficult, so it is better to use mouse, I think.
So it is better to use cell from mouse.
Cell from mouse or human.
Okay.
It is not better to use rat cell.
Okay. And according to your expertise, which one is the maybe the easiest to develop that? I mean, is it from the blood or maybe
in the mouse?
From the mouse or bone marrow or
no.
adipose.
In the mouse is tail fibroblast is commonly used, and also MEF, mouse embryonic fibroblast. Mouse embryonic fibroblast is much better because very efficient. If you can get the embryonic fibroblast, it is easy to generate the embryonic fibroblast.
Yes. Okay. Thank you very much.
Okay. Thank you very much, Dr. Dewi. Okay. So unfortunately, the time is running out. Maybe you have some the closing statement, Dr. Yuasa or Dr. Wawaimuli, or maybe Prof. Asmarina. Is there any closing statement here?
I think from me is enough. So it is already answered your question, and then thank you very much for the participation or for all the participants for the nice question. Thank you.
Okay, thank you very much, Dr. Wawaimuli. Prof. Asmarina, maybe is there any closing statement? You can unmute yes.
Yeah, on behalf of our study program, doctoral program in biomedical sciences, we very thanks for your lecture today, especially for our guest, Professor Shinsuke Yuasa, for nice presentation, nice knowledge for our student and our staff. And also, of course, to our staff, Dr. Wawaimuli, as also speaker in this webinar today. Yeah, for Professor Yuasa Sensei, we will hope that we can make further collaboration as a supervisor for our student, as well also for research collaboration. I think here we have some staff here, Dr. Dewi Sukmawati. I think she is also research in cardiology, and also, of course, Dr. Wawaimuli also, maybe for the next activities, we can joint supervision for our student.
Okay, thank you very much, Prof. Asmarina. Okay, so finally, we come to the end of the session. So thank you very much, Prof. Asmarina, and thank you very much, Dr. Yuasa, Dr. Wawaimuli, and other honorable participants. So we hope we can meet again in the next sessions, maybe in the lecture or something else. Okay, maybe I can present the meeting to the MC, Dr. Oka. So please, if you want to close the meeting. So for me, thank you very much for the meeting. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Jeffy, Prof. Yuasa, and Dr. Wawaimuli for the amazing session. Before